JBEI is super interested in biofuels, and they're interested in organisms that are able to make these biofuels and able to break down some of the precursor molecules that go into making the biofuels. People are so excited about CRISPR and Cas9 because for the first time in a little while, we have a tool that enables us to edit cells directly in living cells. You don't have to take the DNA out and then put it back in and reboot the cell. You have a chance to make a markerless edit to a cell that is living and see the results almost instantly. We haven't had these kinds of tools for plants, for human cells, or even for bacteria. When you have Cas9 available, editing plant genomes to address hypotheses for how to make plants easier to break down and turn into biofuel becomes a lot easier. Cas9 is a part of the complex that CRISPR uses to fight off the virus. CRISPR stands for clustered regularly interspaced repeats, and there are sequences of DNA that we've seen in genomes from organisms like bacteria and archaea since the 80s. Nobody knew what they did until recently. It turned out they're part of an adaptive immune system that bacteria have, which kind of blew everybody's minds. It turns out that bacteria can remember when viruses attack them. They remember a piece of the virus's DNA and they insert it into their CRISPR. They write a little piece of the virus into their own DNA. Then they become somewhat immune to the virus. When the virus reappears, they recognize it and they can fight it. The best part of this adaptive immunity is that they can pass it on to their daughter cells. So our plant people are very excited to use Cas9 to help address some of the challenges of using feedstocks to make biofuel. If you can use Cas9 and quickly edit plant genomes, you can very quickly address hypotheses in how plants grow. If you can use Cas9 so efficiently in yeast, you can very quickly try to make yeast a better host for making biofuels. Cas9 and CRISPR offer us a chance to accelerate our pace of genomic editing so that we can continue to address some of the most pressing problems in our day and age. The creation of biofuel, remediation of pollution in the environment, and the creation of new antibiotics and biomanufacturing, things that bacteria and plants can make but they don't make very much of. Potentially with CRISPR-Cas9, we can make a lot more of them a lot faster and address some of the real pressing problems that our planet is facing.